Welcome to episode 3 of the Physiology 225 podcast. I'm Dr. Jim Davis. Today we're unpacking cell-to-cell communication, which is how your cells send and receive messages to coordinate every heartbeat, breath, and thought. So what is cell-to-cell communication? Picture an orchestra with no conductor. It would be total chaos. In your body, cells act like musicians and chemical signals are their sheet music. Cell-to-cell communication ensures every cell plays in harmony, regulating functions from immune defense to muscle contraction. Every signaling event involves 4 players. First, you've got the chemical messenger. This is the letter carrying information. Examples include hormones such as insulin, neurotransmitters such as acetylcholine, and local mediators such as histamine. Then there's the receptor. This is the mailbox on or in the target cell with a shape that fits its messenger. Think of it as a key, like a key in a keyhole. When the messenger binds, this activates the receptor. Now, next we have transduction. In our introductory course, most receptors are ion channels, or most receptors that we'll talk about are ion channels. When the messenger binds, a channel will open, and ions such as sodium or calcium will flow, which changes the cell's electrical state. Then finally, there's the target cell response. This is the immediate outcome of ion movement, such as depolarization in a muscle cell or secretion in an endocrine cell. Now, cells communicate over different distances with different speeds. There are 5 modes that we'll mention, but we're only going to focus on a couple of them. So first, we have endocrine signaling, and this is one that we'll talk a lot about. This is when hormones travel via the bloodstream to distant targets. An example of this would be insulin, which is released by pancreatic beta cells, enters the circulation and binds to receptors on muscle cells and liver. Then there's paracrine signaling. In this case, messengers diffuse through interstitial fluids to neighboring cells. An example of this would be histamine released by mast cells in allergic response, which acts on nearby blood vessels, which causes vasodilation and increased permeability. And we'll talk a little bit about this one when we get to our urinary system. Then we have synaptic signaling. This is a specialized paracrine mode where neurons release neurotransmitter across the synaptic cleft. It's ultra-fast and highly targeted. An example of this would be a motor neuron releasing acetylcholine onto a skeletal muscle fiber at the neuromuscular junction. This will ultimately lead to skeletal muscle fiber contraction. And then finally, next we have autocrine signaling. And this is when cells respond to signals they themselves secrete. This is something that's common in immune regulation and development, and we'll touch on this just a little bit. An example of this would be a T lymphocyte secreting something called interleukin-2, which binds to its own interleukin-2 receptors to promote proliferation. Okay, and another one that exists but we're not really going to talk about is juxtacrine signaling. This requires direct contact. This is when a membrane-bound ligand or chemical on one cell interacts with a receptor on an adjacent cell. An example of this is something called notch signaling in developing tissues, where notch ligands on one cell bind notch receptors on its neighbor. Okay, so you've got endocrine, paracrine, synaptic, juxtacrine, and autocrine. So let's walk through acetylcholine at the neuromuscular junction. So first you have the release. An action potential, which we'll talk about later on in the semester, reaches the motor neuron terminal. This causes vesicles to fuse and release acetylcholine into what we call the synaptic cleft, which is the gap in between the neuron and the muscle cell. Acetylcholine will then bind to acetylcholine receptors on the muscle cell, specifically on the muscle fiber membrane. Transduction occurs because these channels open and sodium rushes in. This causes something called depolarization. And then the response is this depolarization triggers a muscle action potential, which leads to contraction. So we can integrate this across systems. So the endocrine coordinates long-term processes like growth and metabolism. Paracrine responses regulate local events such as inflammation and tissue repair. Synaptic communication enables millisecond speed responses like reflexes and voluntary movement. And then autocrine and juxtacrine fine-tune immune cell activation and guide cell differentiation during development. Together, these modes ensure your cells communicate effectively across distances and timescales, from the single synapse to the whole body hormonal network. Disruptions in signaling underlie many diseases. For example, diabetes results from impaired insulin signaling, which is a form of endocrine signaling. Autoimmune disorders can involve faulty paracrine or autocrine loops. Neurodegenerative diseases often impair synaptic communication. And cancer can hijack juxtacrine pathways to promote uncontrolled growth. Understanding these modes is the foundation for both diagnosing and treating such conditions. In our next episode, we'll switch gears and talk about the cell membrane, its structure, fluidity, and how it underlies both gradients and communications. And so that's it with cell-to-cell communication. Have a great day.